Hi there. Today we are going to talk about meiosis. We're starting our heredity unit, and in order to understand heredity, we need to understand meiosis. Meiosis is the foundation to understanding heredity. So we're going to take a few minutes to talk about some vocabulary terms that are important for heredity and for meiosis, and then we're going to talk about the process of meiosis. When we look at reproduction and heredity, we are looking at passing genes down from one generation to the next. And there are two ways that genes are passed from generation to generation. And so we're looking at reproduction either asexually or sexually. So asexual reproduction is when you've got one parent and the parent makes a clone of itself. And so you have got one parent and it's producing exact copies, genetically identical copies, usually a result of mitosis or binary fission if you're talking about prokaryotic cells. So cell division, one parent creating genetically identical offspring. So they're making clones. The other type of reproduction is sexual reproduction, where you've got two parents and they are taking some of their genes and combining it with the partner's genes, and you are creating new and different offspring as a result of sexual reproduction. So two parents combining some of their genes to create a new and different organism. When we're looking at humans and human heredity, we are talking about sexual reproduction. So that's where meiosis comes into play, is with sexual reproduction. The key to understanding meiosis and human heredity is understanding homologous chromosomes. So homologous chromosomes are going to be chromosomes that have a similar size and shape and genetic information. So they've got the same genes, even though the genes might be different versions and different alleles. The gene, let's say for eye color, is going to be at the same location on the same chromosome. So those homologous chromosomes are going to be the same size, the same shape, carry the same genes. One of those you get from your mom, one of them you get from your dad. So those homologous chromosomes are going to be the pairs with each other. So they're going to pair up in meiosis. So just a little bit of vocabulary to make sure that everybody understands and is on the same page. We've got different types of cells in our body. Most of our cells are going to be somatic cells, and they're going to have two sets of chromosomes. So humans are going to have two sets of 23. So two pairs of 23 chromosomes, so 2 times 23 would be 46. So most of our cells in our body have 46 chromosomes. The other type of cells are our gametes, which would be our sex cells. So we've got our eggs and our sperm. Each of those are going to have half the number of chromosomes. So eggs and sperm are going to only have 23 chromosomes. And the reason why is so that we can take our 23 chromosomes and combine it with somebody else's 23 chromosomes to make a full set, 46 chromosomes in the baby. So we have to reduce our chromosome number in half to make those gametes, so we can combine our half with somebody else's half to make a whole baby. When we're going through either mitosis or meiosis, before the cells can divide, the DNA will replicate. So those chromosomes duplicate. So here on the left, we've got two chromosomes. So these are going to be homologous chromosomes. So let's say one's from mom and one's from dad. Before they go through meiosis, they replicate or duplicate. And so here on the right, we've got two homologous chromosomes that have duplicated. And they're still counted as a chromosome because those two sister chromatids are still attached together at the centromere. So chromosome number 1 from mom, chromosome number 2 from dad. Those are going to pair up in meiosis and then separate into different daughter cells. And that is kind of the key to meiosis, is those homologous chromosomes pairing up. When they pair up, they form what's called a tetrad. because tetrad, because you've got 4 pieces of DNA, but it's really 2 duplicated chromosomes coming together and pairing up. When we look at a karyotype is going to be a picture of all of the chromosomes in your cell. So when they're coiling up before they divide, if you were to take a picture of all the coiled up chromosomes, cut them all out and line them up, biggest to smallest, that would be your karyotype. So chromosome number 1 is the biggest chromosome, and chromosome number 2 is the next biggest, and so forth. And so you arrange them biggest to smallest. Except for the sex chromosomes at the end, we would have 2 X chromosomes if it's a female and an X and a Y chromosome if it is a male. So chromosomes 1 through 22 are called autosomes, and then our sex chromosomes are our sex chromosomes. So that's a karyotype, a picture of all the chromosomes. And from the karyotype, you can tell if you've got a male or a female individual. And then you can also see if there are any chromosomal abnormalities, extra chromosomes, missing chromosomes, chromosomes that are in the wrong location, pieces that have translocated and moved somewhere else. So we can see chromosomal abnormalities from a karyotype. Again, when we're looking at those homologous chromosomes, you get one from mom and one from dad. Every somatic cell in our body is going to have 2 sets of chromosomes. So they're going to have 2 pairs, and those pairs are going to be homologous to each other. When a cell has 2 sets of chromosomes, it is said to be diploid, or 2N. The N is for the number of chromosomes in a set. And then so 2N would be 2 sets of chromosomes. The N number is going to be different from one species to another, which is why we use 2N for diploid. For humans, the N number is 23, so 2 times 23 gives us 46 chromosomes. When a cell is haploid, like a gamete, that's going to have half the number of chromosomes. It's going to have 1 set of chromosomes, so we say haploid is just an N number of chromosomes. So haploid, half, diploid, 2 sets. And then again, we've got sex chromosomes and we've got autosomes. I mentioned this with the karyotype. Our sex chromosomes are our X and our Y. Females would have XX and males would be XY. The rest of the chromosomes, chromosome pairs 1 through 22, are called autosomes. So just some vocabulary there. So here is a cell that has an N number of 3. So we've got 6 chromosomes, so 2 times 3 is 6. And then these big ones here would be homologous, so they would pair up in meiosis. These 2 medium-sized ones, again, homologous, so they would pair up in meiosis, and these 2 little ones also homologous. So the red ones are maternal, coming from mom, and the blue ones are paternal, coming from dad. So again, gametes are going to be haploid. They would have 1 set of chromosomes. So for humans, the chromosome number would be 23 in our gametes. When the egg and the sperm come together, that process is called fertilization. And so an egg and a sperm fertilized, and a fertilized egg, rather, would be called a zygote. And that's going to have a set of chromosomes from mom and a set of chromosomes from dad, so a set from the egg and a set from the sperm. That will then go through mitotic cell division, so cell division through mitosis, and it would divide and divide and divide, and so it's going to develop through mitotic cell division until it forms an organism, which would then continue doing mitosis to develop into an adult. Meiosis, again, is the production of those gametes or sex cells, and so it is going to have 2 divisions, and it will result in 4 daughter cells after it divides twice. So it's going to divide twice because we're cutting the chromosome number in half, and the DNA has duplicated prior to cell division. So it divides once in meiosis 1 to reduce the chromosome number in half, and then meiosis 2, it is dividing or separating the sister chromatids. Again, meiosis has 2 divisions. The first division is called meiosis 1, where the homologous chromosomes separate. In the second division, meiosis II, that's when the sister chromatids separate. So a lot of similarities between meiosis and mitosis, but there are key differences. And the biggest difference is that in meiosis 1, the homologous chromosomes will pair up, line up, and separate. So you definitely want to pay attention, mostly with meiosis, to what is going on with the chromosomes, because that will help you to understand what is happening in genetics with the heredity and the alleles when we get to that part. So similar to mitosis in interphase, the S phase of interphase, that's where the chromosomes duplicate, centriole pairs duplicate, all of that is going to be the same. But then when we get into meiosis, the stages are going to be a little bit different. So if we look here in this diagram, in prophase 1, that is where your homologous chromosomes will pair up, and that's also where crossing over can take place as well. And then they're going to line up in metaphase 1, and they're going to line up independently from each other. And we'll talk more about this when we talk about heredity, but we've got a pair of red and blue chromosomes. And this pair here, whether the blue or the red is on top, is completely separate and independent from the next pair. And same thing from the next pair. So every single pair is going to line up independently from each other, which means every single time the cell goes through meiosis, they're going to line up differently. So it's going to be a random way they line up. So it's not like all the blue ones are always on top and the red ones are always on the bottom. It's always going to be mixed up. So you're mixing up the orientation and the arrangement of how they separate, which is one reason why we get such differences in our gametes every single time. So we've got crossing over, which I'll talk about what that is in a minute, happening in prophase 1. We've got independent assortment in metaphase 1, where they're lining up. And then in anaphase 1, you've got the homologous chromosomes separating. So this half of these chromosomes, half of them are going to go to one side of the cell, and the other half are going to go to the other side of the cell. So all those homologous chromosomes are going to separate, and half go to one cell and the other half go to the other. And then in telophase 1, cytokinesis is going to occur. And now we have two daughter cells that have half the number of chromosomes, but they're duplicated, so they have to divide again. So meiosis 2 is similar to mitosis in the fact that the sister chromatids, the chromosomes line up in the middle, and then the sister chromatids get pulled apart and separate and go into different daughter cells. So then after cytokinesis occurs in telophase 2, then you have 4 daughter cells. Each of them have half the number of chromosomes, and they're all different from each other. They're different from each other because some of them have blue chromosomes, some of them have red chromosomes. So it's a mix of blue and red. The mix is going to be different. And then also we've got crossing over occurred in meiosis 1 in prophase 1, which has also mixed up the genes a little bit. So every single cell has half the number of chromosomes, and they're all different from each other, and certainly different from the original parent cell. So that kind of is meiosis in a nutshell. 2 divisions. The first is separating the homologous chromosomes. The second is separating the sister chromatids, all of which is reducing the chromosome number in half, and it is creating 4 haploid daughter cells in the end that are all different from each other. Okay. So again, you want to pay attention to what is happening with the chromosomes. So prophase 1, the homologous chromosomes pair up to form tetrads. Then the homologous chromosomes the pairs will line up across the middle of the cell in metaphase 1. And then anaphase 1, those homologous chromosomes are getting pulled apart and moving towards opposite ends of the cell. And then telophase 1, those homologous chromosomes are now at the opposite ends of the cell, so cytokinesis can occur, cutting the chromosome number in half. Then, of course, they have to divide again, meiosis 2. And so prophase 1, the centriole pairs are going to duplicate again and move to opposite ends of the cell so that they can pull the sister chromatids apart. So chromosomes then in metaphase 2 line up in the middle of the cell. And so this is where it is similar to mitosis. And then anaphase 1, the sister chromatids separate and move to opposite ends of the cell. So again, the kinetochore fibers shorten, pulling the sister chromatids apart. Telophase 2, cytokinesis occurs again. Chromosomes start to uncoil, the nuclear membrane forms again. We have now 2 haploid daughter cells, each of which are different from each other. Then this diagram kind of sums up the whole process. So in interphase, you're starting off with your pairs of homologous chromosomes. So in this simplified diagram, you have one maternal and one paternal chromosome. Interphase, in the S phase, they duplicate. So they are sister chromatids attached together at the centromere. Then in meiosis 1, those homologous chromosomes separate and go into separate daughter cells. And then meiosis 2, each of those have sister chromatids. They get separated and go into separate daughter cells. So we all have haploid daughter cells with half the number of chromosomes, 4 daughter cells that are different from the parent cells. So comparing meiosis to mitosis real quick. Mitosis, you are creating genetically identical daughter cells. And the chromosomes, as you can see in metaphase, line up single file. But in meiosis 1, you've got the pairs. So it's more like couples lining up instead of single file lining up. So homologous chromosomes separate, and in meiosis 2, there is a second division separating the sister chromatids. Okay, I mentioned in prophase 1 that crossing over sometimes occurs. Crossing over is when the chromosomes, when the homologous chromosome pairs, when they pair up, they get really, really close together. And so sometimes the DNA from one chromosome will swap places with the DNA from the other chromosome. So basically, a little bit of the blue breaks off and attaches onto the red, and a little bit of the red breaks off and attaches onto the blue. So the DNA will swap. This adds diversity and variation to the genes, which is also making individuals that are different from each other. So this is a way that we can add variation to the population. So we're getting different gametes every single time we make gametes. Here's just another diagram showing you the crossing over taking place. So here it took place in 2 locations. So we've got a little bit of red and on the top and the bottom of this sister chromatid, and a little bit of blue on the top and the bottom of this red chromosome. So now if you look at the bottom, the daughter cells, they're all different from each other. So that's why when we look at brothers and sisters and siblings, they have, even though they come from the same parents, the genes have been mixed up in crossing over and independent assortment. So we're adding variation to the population that way. Just another diagram showing you crossing over taking place. So to sum up, meiosis is making gametes or sex cells. Each gamete has half the number of chromosomes, and you get 4 different daughter cells being formed. The process of making gametes is called gametogenesis. So meiosis in males and females, slightly different. So gametogenesis is the production of those gametes, and there are some differences between males and females. So sperm are going to be movable or motile, so they've got a little flagella on them that can help them travel to get to the egg to fertilize it. Eggs are going to be larger, and they're going to carry out their function within the female. So the purpose and the production process is slightly different. So just going to talk a little bit about it, to talk a little bit about the differences in meiosis in males versus females. So looking real quick at spermatogenesis, that is, of course, the production of your sperm. And when we're doing spermatogenesis, we are making 4 sperm. oogenesis is the process of making eggs in the females. It does take generations, years, sorry, to make the eggs, and it does happen over decades. So it's a slightly different process. So if we look at how spermatogenesis differs from oogenesis, making sperm versus making eggs, there's a few ways that they differ. So all four products of meiosis develop into sperm, while only one of the four becomes an egg. And we'll take a quick look at that in a second. Spermatogenesis occurs throughout adolescence and adulthood, so it's a constant process of making sperm all the time. And sperm are produced continuously without the prolonged interruptions of oogenesis, and I'll talk about what that is in a second. So looking first at spermatogenesis, you can see these cells here are the ones that are going to develop and go through meiosis to develop into the sperms. So basically, you're looking at stem cells that will divide until you form the spermatocyte, which is what's going to then go through meiosis. So each primary spermatocyte is going to go through meiosis and produce 4 daughter cells that are haploid, and then produce this 4 sperm. When we look at oogenesis, again, we've got germ cells or stem cells that will divide into cells that are then going to go through meiosis. But with females, we have a pause in between in the process. So it's going to start meiosis in development. So in the fetus, some of these cells are going to go through the first part of meiosis, and then they're going to pause. And then every month, one of them is going to continue with meiosis, and it's going to release and it's going to divide. And as it divides, so in ovulation, one egg is going to be released, and it will finish the first division of meiosis, and it's going to release one polar body and one cell that will become an egg. So cell division, or cytokinesis rather, is uneven. So the egg is much bigger than the first polar body, but the DNA, it splits, the homologous chromosomes divide. And then if a sperm fertilizes the egg, then it will continue through meiosis. It will do the second meiotic cell division. So it will go through meiosis 2, and a second polar body will come out, and the fertilized egg will then, the nucleus will merge with the nucleus of the sperm, and then you've got a fertilized egg. So basically, really what's happening is you're starting meiosis, you're pausing meiosis, you're starting meiosis again, you're pausing meiosis, and you're starting meiosis again in the process. So you only get one egg out of one primary oocyte, as opposed to getting 4 like you get in spermatogenesis. So that's kind of the big difference between sperm and egg, is one of those is going to develop into a fertilized egg. The rest of them are going to be polar bodies, which are support cells. So a little bit different process. So it's important to understand that you're really only getting one egg from one original parent cell in that process. Okay. And then the last thing that we're going to talk about real quick is if the chromosomes don't separate correctly. And if that happens, that is called non-disjunction. And that's going to lead to the wrong number of chromosomes in the gametes. And then if that gamete is fertilized, then the offspring will have the wrong number of chromosomes. And that will lead to genetic disorders, which we will talk about later. But non-disjunction can happen either in meiosis 1 or it could happen in meiosis 2. And in either case, you're going to get the wrong number of chromosomes. So if non-disjunction occurs in meiosis 1, then both of these homologous chromosomes end up in that first daughter cell. and you now have an extra chromosome in this cell and a missing chromosome in that cell. And then when it divides the second time, you've got two cells with an extra chromosome and two cells with a missing chromosome. If non-disjunction occurs in meiosis 2, then the homologous chromosomes separate correctly in the first division, but the sister chromatids don't separate correctly in the second division, putting an extra chromosome in this cell, a missing chromosome in that cell, and then these two are perfectly fine. So if non-disjunction occurs in meiosis 1, then all of your gametes have the wrong number of chromosomes. If non-disjunction occurs in meiosis 2, then half of your cells have the wrong number of chromosomes and half of your cells have the correct number of chromosomes. So non-disjunction is when the chromosomes do not separate correctly, creating cells, gametes with extra or missing chromosomes. Okay, what we're going to do next is take a quick look at this BioFlix video, and then that's it for meiosis. So let's take a look at this video.
Humans produce gametes, eggs and sperm, through the process of meiosis. Here, we'll follow the production of male gametes by focusing on this cell as it goes through meiosis. Let's begin in the nucleus, where genetic information is stored in chromosomes. Most of a person's cells are diploid, with two sets of chromosomes. One set is from their mother, shown here in red, and the other set is from their father, shown in blue. Each maternal chromosome has a corresponding paternal chromosome. These matched pairs are called homologous chromosomes. During interphase, chromosomes are duplicated. Each chromosome now consists of two identical copies called sister chromatids. Zooming in, we see that each sister chromatid is made up of DNA wound around histone proteins. Each strand coils up into a tight helical fiber. As meiosis begins, a spindle forms and duplicated centrosomes start to migrate toward opposite poles of the cell. Back in the nucleus, the chromosomes are condensing. In meiosis, homologous chromosomes stick together in pairs. The close association of homologous chromosomes allows segments of non-sister chromatids to trade places. This recombination of maternal and paternal genetic material is a key feature of meiosis. After the spindle forms and the nuclear envelope breaks down, microtubules from opposite poles attach to each chromosome of the homologous pair, resulting in a tug-of-war. At metaphase 1, the chromosome pairs are positioned in the middle of the cell. The next stage begins when homologous chromosomes separate from each other and move toward opposite poles. Each chromosome still consists of two sister chromatids. This cell began meiosis with 46 chromosomes, but each daughter cell now has only 23 chromosomes. In meiosis 2, microtubules from opposite poles attach to the chromosomes, which then move to the center of the cell. Next, the sister chromatids separate, becoming full-fledged chromosomes that move to opposite poles. Nuclear envelopes reform, and each daughter cell divides into two cells. We started with a single diploid cell, and now that meiosis is complete, we have 4 haploid cells with a single set of chromosomes. These haploid cells mature into gametes that can contribute to the next generation.
Okay, hope that was helpful, explaining a little bit about meiosis and some meiosis and genetics vocabulary. Catch you next time.